In this video, we're going to take a look at how to interpret figures in a primary research article. The goal is for you to be able to draw conclusions based on the data in the figures that are found in these primary research articles, and then to predict the outcome of experiments related to figures based on how the data come out. So one type of figure that occurs in many molecular biology papers is a so-called Western blot that's used to measure the abundance of specific proteins. So let's take a look at one of these example figures here. So in this case, the darkness of the band indicates the abundance of that protein, how much of that protein was present in the sample, whether it was cells or some tissue from humans or mice or whatever the sample is. The authors here are trying to measure the amount of that protein in the sample, and that's indicated by the intensity of this band. And the way this is done is by separating all of the proteins in the sample out on a gel, where the larger proteins are higher on the gel and the smaller proteins run faster and are found on the bottom of the gel. Then they usually focus on the particular molecular weight region of interest. So in this case, these authors were looking for the hexokinase 2 protein in these samples. And so they focused in on the range of molecular weights around hexokinase 2. So the whole gel would be much bigger than this, but they're only showing us a part of that gel where hexokinase 2 is expected to appear. And then they use an antibody that specifically recognizes hexokinase 2. Now, sometimes there'll be some other bands that are recognized that are non-specific, we say, but in most of the published cases, they've found an antibody. Like in this case, it looks like it's very specific for hexokinase 2. So you don't see any other bands that are recognized with this antibody, at least in the molecular weight region that they're showing us. And we just see this one band going across at different abundances, different intensities. Presumably that's all representing different amounts of hexokinase 2 in these different samples. So if we look at the first column here, what's happening? So we see there's one treatment condition and a certain low level, kind of faint band associated with hexokinase 2. So that means that in these samples that were treated with the LAC-Z adenovirus, they had only a small amount of hexokinase 2, the amount indicated by this faint band. Now, what if they made a mistake when they prepared those samples and there was nothing in the sample? Basically, all the proteins somehow were lost from the sample when they were purifying it. So then you would also maybe have a very faint band or no band at all. So to control for that, to check for that, it's common practice to also, in the same sample, run a parallel analysis where you look for another protein of interest that you do expect to be present, and that ideally is not changing between the different samples. This is called a loading control, meaning that they can show that they've loaded or put equal amounts of protein samples overall, total protein, into each of these wells and the gel, each of these columns, each of these lanes. And then it's the particular treatment they've done here that causes the variation in hexokinase 2, not the amount of protein they loaded on the gel or how well they were able to break open the cells and collect the protein, or some other kind of technical aspect of the experiment. So in this case, they looked at two so-called loading controls. They looked at actin, which is a cytoskeletal protein that's commonly used. It's present in all cells. It doesn't really vary too much under different conditions. And so they see, yes, there's a band, a single band for actin. It's present at about the same intensity in all 4 samples. So it means that that actin protein is equally abundant in the samples. They prepared the same amount of protein roughly in all of these samples and loaded equal amounts of it onto the gel. Now they have another control here, hexokinase 1. That's a protein, again, that they don't expect to change across the samples, but they know that it's very similar to the one they are interested in, hexokinase 2. Now why do they want to test for changes in hexokinase 1 across the samples? Well, you could argue, oh sure, actin is not changing between your samples, but all of the enzymes involved in glycolysis are changing. And so this difference in hexokinase 2, it really just reflects differences in all of the enzymes of glycolysis. So they want to have another protein that's as similar as possible to hexokinase 2, but that they don't expect to be different between the samples. So that shows that whatever changes they see in hexokinase 2 abundance are pretty specific to hexokinase 2. Maybe just that protein and no other proteins are changing. Now there might be a few other ones they haven't checked for that are changing, but in general it would mean that it's not a totally non-specific effect, meaning it's not like lots and lots of proteins are changing and only the cytoskeletal ones are not changing. So in any case, they have a general loading control here, actin, so they see about equal abundance in each lane. And then they have a specificity control for loading and specificity of the changes they see. This specificity control is hexokinase 1. Now what about hexokinase 2? So now the key question is, knowing that they have equal amount of protein and good preparation of all their samples based on these controls, what's happening with hexokinase 2 in their samples? Well, let's take them one lane at a time. So again, in the first lane, under the control conditions, they treat the cells with adenovirus lac-Z, and they have only a little bit of hexokinase 2 protein, this kind of faint band that means a little bit of protein in that sample. Now in the conditions where they treated the cells with the Cre adenovirus, they see that hexokinase 2 band is gone. If we compare lac-Z, ad lac-Z with ad Cre, you see that faint band that was there is no longer there in the ad Cre treatment. Now they also note on the bottom that these 2 samples both contained a dominant negative p53. Why are they telling us that? Well, they're telling us that because they also want to tell us that the next 2 samples they looked at on the right had not only dominant negative p53, but also had HRAS V12 mutant, which is an oncogenic mutant of the RAS protein. And so they're going to be comparing the effect of introducing HRAS V12 into these cells. But they want you to know that was done in the background or in the presence of all of the samples having dominant negative p53. Now they could have just mentioned that in the text, but it's common if in this case where they're doing a specific intervention to all the samples just to show that clearly in the figure. So you know that all 4 of these samples have dominant negative p53. Okay, so again, comparing the first 2 lanes, we see that the adenovirus Cre treatment caused hexokinase 2 to disappear, as you would expect if you read more about this experiment in that paper. Well, what about the next 2 lanes? Well, in the next 2 lanes, they're treating these cells with this mutant HRAS protein. So it's got a V12 mutation, which makes it mostly active all the time and makes it oncogenic. And so these 2 samples, in response to that HRAS V12, show a change in hexokinase 2 level. Now, we know that this is something specific to hexokinase 2 because they showed us these nice controls. Hexokinase 1 is about the same in all the samples. Actin is about the same. So HRAS V12, when introduced into these cells, is causing more of the hexokinase 2 protein to be present. Right, so if we compare, usually with these kind of multi-sample experiments, we have to compare, we have to think about which are the comparisons that are relevant. And usually it helps to start with the first lane, because they usually set it up logically. So you can start with the first lane and compare to that. So in this case, for example, if we just infect the cells with the LAC-Z adenovirus, that's really a controlled treatment, then we see there's a low level of hexokinase 2. If we add in HRAS V12, we get a lot more hexokinase 2 protein, comparing the first and the third lanes there. So the effect of HRAS V12 is to cause more hexokinase 2 protein to appear in the cells. Now, if they do this AdCre treatment, then it causes the loss of hexokinase 2 normally, and then in the presence of the HRAS V12 mutant, it still causes a significant loss of the hexokinase 2 protein. So we're seeing that again. There's two comparisons here: add LAC-Z to AdCre, and then the effect of without or with HRAS V12. Okay, so that's how you would read these kind of figures. You would look to see, well, what's the loading control? Does that look similar in all of the lanes? And then what are the different treatments they're comparing? Now, really, the key thing they were trying to do here was, again, there was two comparisons. They want to know, what is the effect of introducing AdCre? Well, in order to know what that effect is specifically of CRE, they need to do another control infection with the adenovirus that doesn't really do anything. And so the LAC-Z is supposed to be an adenovirus that doesn't really do anything. It's just to control for the process of exposing cells to an adenovirus, but without any CRE being expressed. So that's the comparison here. What's the effect of CRE, introducing CRE into the cells, CRE versus just a control adenovirus. And then if you compare the left two lanes to the right two lanes, what's the effect of introducing the oncogenic HRAS V12, specifically in terms of making more hexokinase 2 protein, comparing the first and the third lanes. So the other major kind of figure you often see in primary research articles is a bar chart. A bar chart where we're just comparing the effect of two different treatments or two different conditions or an intervention on some process of interest. So let's look at an example here. Here, these authors were looking at what happens when they remove hexokinase 2 from cells. So these are RAS cells that either have hexokinase 2 on the left. Now it's set up in a way that it can be deleted with the AdCre treatment. FF is the floxed allele, so it means that this hexokinase 2 gene they've introduced is ready to be removed by treatment with AdCre. When they treat with AdCre, they generate the hexokinase 2 null cells that don't minus, meaning they don't have hexokinase 2 anymore. That gene has been deleted. That's the effect of CRE, turns them from floxed to null or to minus. But the genetics are not so important here. But I mean, this could be apples and oranges. It could be any two things. In terms of reading the figure, there's treatment A and treatment B. In this case, it's the presence of hexokinase 2, or after hexokinase 2 is deleted, they want to see what is the effect on a particular activity or event or something that's measurable in the cells. In this case, they're measuring total hexokinase activity. So they want to know, when we delete hexokinase 2, how much of the total hexokinase activity, which is due to hexokinase 1, 2, 3, 4, etc., how much of that total hexokinase activity is removed? So in other words, what fraction of the total is hexokinase 2 actually normally responsible for? And so in this case, it looks like they've set this to be normalized to 100% hexokinase activity. So that whatever number they measured for the activity using their assay, they set that equal to 100%. And you can see it looks like they measured this a few times, and then they have an error bar which tells you how variable was this measurement. As the error bars are larger, it means that with repeated measurements you're going to have more variability in the number. So in other words, when they measure this, it didn't come out at exactly 100. It might have been 98, 103, or on this scale. So there's some error associated with that measurement, which is what the error bar there is telling us. Now when they deleted hexokinase 2 and they're looking at the null cells, now they have a lot less, somewhere down in the 30-35% of total hexokinase activity remains compared to the starting situation where it was set at 100%. And again, there's some error associated with this measurement, so they're showing an error bar, and that gives us an idea of how variable the measurement was. Now these 3 asterisks indicate that when they did a statistical test to compare the significance of these differences given the error bars associated with each measurement, this is unlikely to be seen by chance. And if you go into the methods in the paper and the details in the legend, you'll see the exact statistical test and the significance of the difference. But usually when you see these 3 asterisks or 2 asterisks, something like that, 1, 2, 3, some number of asterisks, sometimes they'll write the p-value in there. It means the probability observing this difference by chance, given the error bars, is very low, less than some percent. And by convention, we usually say if it's less than 5% or 1% chance that you would see this difference, given the error bars you're seeing, then that's probably a real difference and a significant difference. That's kind of the convention in these kinds of papers. So in this case, they're saying when we have the hexokinase 2 null cells, they have only about 30-35% of the total hexokinase activity that's present normally in cells that have hexokinase 2. There's some error associated with these measurements, but the error is small enough that we can say this difference is very likely to be a genuine difference and not an error due to measurement. Let's look at another example. You can see you can do the same kind of comparison for whatever readout or effect that you want. So again, let's say you want to compare what happens when you remove hexokinase 2. You have the hexokinase 2 null cells with the cells that have hexokinase 2 floxed on glucose consumed. So how much glucose are these cells consuming? Again, looks like they've set this to 100%. So they measured the amount of glucose consumed, and that's whatever number they came up with, amount of glucose consumed, they set that equal to 100%. Again, there's some error associated with that measurement. Then they checked what's the glucose consumed in the hexokinase 2 null cells. Looks like around 50-55% of the level that was consumed in the cells that have hexokinase 2. So there's a difference there, and there's some error associated with each measurement, but they did their statistical test and they said, aha, this difference is very unlikely to be seen by chance, so this is a significant difference in the amount of glucose consumed under these 2 different conditions. And then again, we can just look at another example. So hexokinase 2 null cells versus the hexokinase 2 flock cells, in other words, the ones that have hexokinase 2. What about tumor size? So the tumor size, the tumors derived from the hexokinase 2 null cells, it's quite a bit smaller. It's 400 or so cubic millimeters versus over 1,500 cubic millimeters. Now in this case, they haven't normalized the value to 100%. They could have done that, but the convention in reporting tumor size in these kind of tumor xenograft experiments is you just report the actual size of the tumor. You don't normalize it, because the actual size of the tumor is important. You want to know if you're dealing with big tumors or little tumors. That changes how they respond to things. So they're showing us the actual size of the tumors. 1,500 cubic millimeters is quite large, extremely large. 400 or 500 is still a decent size, although it's obviously a lot smaller than 1600. And again, there's some error, a little more error it looks like associated with these measurements, but still, based on the test they did, the tumor size in the HK2 null cells is significantly less. In other words, unlikely to be due to just random error of the measurement, but a genuine difference in the tumor size under these two conditions. Now, sometimes, quite commonly really, you'll see that there are multiple comparisons performed in the same figure. So usually it's at least two different kinds of comparisons. So here again, let's look at tumor size. And usually don't get thrown off or intimidated by the figure. You just need to work your way through it, starting on the left and thinking about what are the different comparisons they're trying to make. So in this case, they're measuring tumor size, and they're starting with just their control sort of baseline conditions. If they have a LAC-Z adenovirus as a control adenovirus that shouldn't really do anything, and they introduce a vector plasmid that shouldn't really do anything, so like two treatments that shouldn't really do anything, then the baseline tumor size they're seeing under those conditions is very large. These tumors grow up and become almost 2,000 cubic millimeters. So you get these big tumors growing. Okay, now let's move to the next bar. So what is the next bar? What's different between these? Well, they're still using the control vector plasmid, so they're not introducing any protein coding gene or cDNA. They're just using a vector as a control. So again, the vector means it shouldn't really do anything. That's just the delivery vehicle for the gene that they're going to test later. So we can ignore that for now. And the treatment they're comparing now is between the AD-LAC-Z and AD-CRE. So now you're looking at what is the effect of this AD-CRE treatment, introducing the CRE recombinase versus just a control adenovirus that shouldn't do anything. And they say, aha, the tumors become quite a bit smaller, less than half the size, 800, 900 cubic millimeters when grown for the same amount of time. Now, because they want to draw our attention to this comparison between the LAC-Z and the AD-CRE treatments in these first two bars, they draw a line and then they show that this comparison meets a certain threshold of statistical significance indicated by the two asterisks. That's a common way to say, yes, these two comparisons are significantly different. Given the error here, we still find this to be unlikely to be due by chance, and this is a genuine difference, a significant difference. So they're saying that when they put in the AD-CRE, they get these smaller tumors. Okay, well, what about the next bar? So the next bar is saying, well, it's a control for the AD-CRE treatment. What they're trying to prove to us in this figure is that when they do the AD-CRE treatment, the smaller tumors are because they've removed hexokinase 2. Well, why do they need to do that? Well, for example, what if the CRE just had some other toxic effect on the cells? Maybe it induced DNA damage, it somehow damaged the cells, and it caused smaller tumors, but it wasn't really due to the loss of CRE, or the loss of hexokinase 2. How could they prove that the effect of CRE in reducing tumor size is due to removing hexokinase 2 from that fluxed allele? Well, the way they could do that is by showing if they treat with AD-CRE and they add back hexokinase 2 in a plasmid, that should reverse the effect of AD-CRE. So in other words, does reintroducing hexokinase 2 into the cells that were treated with AD-CRE, does that reverse the effects of AD-CRE? If AD-CRE was just damaging the cells in some other way that had nothing to do with hexokinase 2, then when you introduce hexokinase 2, it wouldn't reverse the effect of AD-CRE. The fact that when they put in hexokinase 2, they now see big tumors again means that most likely the effect of AdCre was through removing hexokinase 2, which we kind of expect that it was, but they're trying to prove this to us in a more definitive way. So now when you have hexokinase 2 re-expressed in these cells, they have big tumors again. Okay, well now again, it's like with all these experiments, you're thinking like the devil's advocate, the skeptic is saying, I don't believe you because what about this, what about that? So now you could say, okay, yeah, but maybe introducing hexokinase 2 had some other weird effect, like the protein was unfolded and just started to gum up the protein degradation machinery, or it had some kind of weird chaperone effect, or some non-specific effect of expressing hexokinase 2. Oh, okay, so what you want to know is when I express hexokinase 2 in these cells to reverse the effects of AdCre, does that depend on the kinase activity of hexokinase 2? It is, after all, a kinase, phosphorylates glucose to make glucose 6-phosphate. Does this tumor size effect of hexokinase 2 expression depend on the kinase activity of hexokinase 2? So they can test that by doing the same rescue experiment, but now with a mutant hexokinase 2 that doesn't have kinase activity. So it's a so-called kinase dead serine to alanine mutant. So the kinase dead mutant, SA mutant, here on the right, should not rescue the cells if the kinase activity of hexokinase 2, in other words, its main normal function that we know about, phosphorylating glucose, is involved in this tumor rescue. And that's what they see when they treat with AdCre and add back the kinase dead hexokinase 2, they get the small tumors. So the mutant hexokinase 2, the kinase dead hexokinase 2, cannot reverse the effects of AdCre. So basically what they've done here is they're showing us that AdCre has one effect of reducing tumor size, and then they're trying to show us the mechanism of that is through removing hexokinase 2, because they can reverse the effects of AdCre by adding back hexokinase 2. So that's really a control. Hexokinase 2 is a control to show how the AdCre is working. And then the kinase dead lane is a control for the control to show how the hexokinase 2 rescue is working. So there's really a lot packed into these figures. And they do each of these comparisons, the statistics, and they show us that the effect of AdCre indeed is significant. The effect of the difference between the rescue here and the original vector is not significant, meaning they've fully rescued the tumor size by hexokinase 2 expression back to effectively what it was in the beginning, and that when they put in the kinase dead mutant, they compare that to the original tumor size. That is significant, showing that the kinase dead cannot rescue. So they need to show us these statistics to convince us that what looks visually like a real difference is statistically different when you run the numbers. Okay, so that's an example of how to interpret figures from a primary research article, and really two main kinds of figures that you'll come across: Western blots, where you're measuring protein abundance, and bar charts, where you're comparing the effect of two different conditions on some property of interest.